Welcome to the deep dive. Today, we're zeroing in on a really fundamental technique in genetics, specifically for complex traits. It's called QTL mapping.
That's right. And we're going even deeper into one specific method, single marker analysis, or SMA.
We've looked at some course materials, transcripts. Basically, we want to unpack what SMA is, how it functions, and maybe surprisingly, where it falls short.
Exactly. What are its strengths and what are the, let's say, hidden weaknesses? Pulling out the key stuff for you.
Okay, maybe a quick refresher first. QTLs, quantitative trait loci. These are spots on the genome influencing traits that vary, like height or yield, not just on-off traits.
And QTL mapping is just the process of finding those spots.
Right. So where does single marker analysis fit in?
Well, the sources frame it as maybe the simplest starting point for finding links between molecular markers, the genotype at one spot, and the trait you're measuring, the phenotype.
So it's like step one in the association part of mapping?
Pretty much, it's about finding that initial signal.
Okay, so the core idea, how does this simple check actually work? What does it do?
It's straightforward, really. SMA looks at all your individuals, divides them up based on their genotype at one single marker.
It's just one marker at a time.
Yep. And then it asks, is the average trait value significantly different between the group with, say, genotype AA and the group with genotype A at that specific marker?
The materials mentioned examples, right? Like mouse body weight?
Yeah, mouse body weight in a backcross population, that means just two possible genotypes per marker. Or maize grain yield in an F2 population where you'd have three genotypes like AA, AA, and A.
And apparently sometimes you can almost just see a difference if you sort the data.
Sometimes, yeah. If the effect is strong enough near that marker, but usually you need stats, a t-test, an f-test, or something called a LOD score, if those show a significant difference in the averages.
Then SMA says.
It suggests there's likely a QTL, a gene affecting the trait, somewhere near that marker. It's a putative QTL, not confirmed, just suggested.
Okay, so statistically, what's going on under the hood when it finds that difference?
Fundamentally, it's using linear regression. It's modeling the relationship between the marker genotype and the trait value.
Simple enough for the two genotype case, like the backcross.
Right. For the backcross, it's basically simple linear regression. You're testing if the difference in the average trait value between the two genotype groups is significantly different from zero. Is there a real slope, a real effect associated with that marker?
And for cases with three genotypes, like the F2 maze?
Then you can use multiple linear regression. This lets you try and tease apart different types of genetic effects potentially linked to that marker, like additive effects versus dominance effects.
Okay, sounds fairly standard so far. Simple method, simple stats.
But here's where the sources give a really big warning. The major catch. Especially clear in populations like the backcross.
What's the catch?
The genetic effect you estimate with SMA, well, it's confounded. It's biased.
Confounded bias. Okay, what does that actually mean for the result? Is it just wrong?
It means the difference you detect isn't purely the effect of the QTL itself. It's mixed up with something else. It's influenced by how far away the marker you're testing is from the actual QTL.
You mean the recombination frequency, the genetic distance?
Exactly. The recombination frequency R between the marker and the true QTL location gets tangled up in the result. The difference you measure is proportional to this calculation involving R and the true additive and dominance effects of the QTL. Something like 1 to R times the difference in genetic effects.
Whoa. So if SMA flags a marker as significant,
You don't actually know why it's significant based on SMA alone. Is it because the marker is genuinely close, small r, to a QTL with a real effect? Or is it maybe further away from a QTL with a huge effect? Or maybe it's just the combination of R and the effects happen to produce a significant number.
so the signal is real, but its meaning is fuzzy.
Very fuzzy. And crucially, the sources stress this. SMA cannot tell if there's one QTL near that marker or if there are actually multiple QTLs linked together in that same region.
Because it just sees the combined effect at the marker location.
Right. And because of all this, it tends to underestimate how many QTLs there really are, and it underestimates their true individual effects.
Okay, let's sum up the pros and cons then. Pros seem clear.
Yeah, pros. It's easy. Simple concept, simple to run. You don't need fancy software. You don't even technically need a genetic map showing where all the markers are relative to each other. You can just test marker by marker.
Simple and accessible. But the cons sound pretty serious.
They are. That confounding is the big one. It means SMA can't resolve multiple linked QTLs. It doesn't give you a precise location for the QTL, just near the marker. It underestimates the effects. And this confounding actually reduces its statistical power to even detect QTLs in the first place. It misses things.
So it's a starting point, maybe, a first pass.
Exactly. It's foundational, accessible, gives you a signal. But you absolutely have to understand its limits.
Yeah.
That confounding bias, the inability to pinpoint locations or untangle linked effects, those are critical limitations.
It really highlights how a simple association signal doesn't always tell the whole story, doesn't it?
Absolutely. You get a flag saying something interesting here, but figuring out the actual genetic architecture, where the QTLs are, what they each do, how they interact, that needs more advanced methods that build on this but go beyond just one marker at a time.
So the signal from SMA is a clue, but interpreting what it really means biologically takes more work and caution.
Precisely. Makes you think, doesn't it?
Yeah.
How often in looking at data do simple correlations hint at something real, but potentially mask a much more complex underlying reality?
A good thought to keep in mind whenever we're looking at associations in data, something to ponder.